So we're going to move on to Tom Farb. So, you know, if I was going to talk about like an archetypal person that we look at in our group, we look at, there you go. See, he's already, people are already clapping for him before he's even said a word. There you go. So we brought the president of his fan club to our event. And, you know, the great thing is that we look at a lot of guys who are multi-time entrepreneurs, and in particular in the healthcare space, because, you know, healthcare is a labyrinthian sort of venture to enter into whenever you're doing it, because you have to know who are the payers, who are the regulators, et cetera, and what's the environment that you're entering into. So it's a lot more complex than just like, you know, selling chat to people all around the globe. Tom Farb is a multi-time entrepreneur, multiple exits. Several of them are just fantastic. He's schooled in AI and machine learning. Some of his ventures have not just been healthcare. And so he's got an interesting healthcare IT product that may revolutionize the way labs approach things. I think there was an issue a couple of years ago where there was like a lab, I don't know if it was in Massachusetts, where they found out that a lot of it's had been contaminated, and they were using it for like police research on genetic testing. Right. And so they had to throw out all the results for like, you know, 20 years.
People who might have gone to jail or should have did not.
Right. So yeah, very interesting. So there's a lot of flaws in the current system for labs that we have. And Tom Farb is going to talk about Thrive Bioscience, what they do, their approach. So Tom, take it away. And I'll pull up your, actually, I'll sit here and I'll do it. One second.
Okay, great. Thank you. So my own background, I've started 11 companies. 5 have had multi-billion-dollar exits, and they've been in AI, life sciences, and within life sciences, diagnostics, devices, and pharmaceuticals. So when I start a company, I know I'm going to be working endless hours. I might as well have a really big impact. It's part of what keeps you going when you face all these various challenges. And so one of the biggest problems that we all face is the lack of medicines and the high failure rate. And I figured out, in part from reading a really excellent article, what a significant portion of the problem is. And the problem is that drug discovery is based on cells and cell culture and cell-based experiments. But amazingly, there is almost no data that is collected about these cells. In fact, a quarter of the time, the cells are mislabeled. So we think we're studying prostate cancer, but we're really studying liver cancer. So Thrive Bioscience is providing for the first time data sets that allow data mining, artificial intelligence, and breakthrough insights. This data has just not been available before. So let me give you some key points about Thrive. So our products are for providing data and analytics, primarily for drug discovery. This is a huge market, the cell-based market. Cell culture is a $29 billion per year market. And it affects almost all aspects of life sciences, all segments. Our products do not require FDA approval. So there's not that kind of binary problem that you have. We sell to all the people who do have binary problems, who do have FDA approval or non-approval. We've sold 15 instruments. We're just at the beginning of revenue. And I'm pleased to say we just got an order for $2 million from one of the largest life science companies in the world. We have significant collaborations with a large pharma in Japan, as well as Harvard Stem Cell Institute, Stanford Stem Cell Core, the Broad Institute, and 5 others. We project sales of $4 million in the next 12 months and growing to 150 million in 5 years. And in the 5 multi-billion-dollar exits that I've had, we've done that same kind of growth in all of them. So it's an experienced team. Previous panelists talked about the importance of the founder. I would add to that founders. It's very hard for one person to do it all and not have somebody to talk to. And not every founder makes it. So we started with three. We now have two, and I have a fantastic co-founder who has made some of the most important inventions and discoveries in genomics. He developed some of the first sequencers, and the sequencers that are sold by Illumina, which is the vast majority of them, were developed by my co-founder. So we have one of these unique areas that's been neglected. Every once in a while, you come across an area that hasn't been digitized, that tools haven't been developed for. And we found that with cell culture labs. They've largely been ignored. And I found the same thing in one of my previous companies, Cytic, where we automated cytology labs, which provide pap smears. And pap smears are the largest single primary screening test in the world. And before Cytic, they were done the same way for 65 years. And Cytic automated that, automated cytology labs, just like we're automating cell culture labs. And as a result, 90% of all pap smears use our instruments. We sold the company for $6 billion to Hologic after taking it public. And this is a very similar situation where there's been limited innovation. And so in order to protect ourselves and the investment that our investors are making and to enhance the exit, we have filed 78 patent applications and 25 have been issued. We've had none that have been denied. And that's really because this area has been neglected, including on the IP side. Our instruments have not been built before. So we've raised $31 million to date, mostly from family offices. The Estee Lauder family, which is invested in a couple of my previous companies and had pretty fantastic returns. They invested in HNC software, where we basically monitor to this day 90% of all credit cards and debit cards using AI. That company, HNC Software, was acquired by Fair Isaac, and a division we spun off as a public company by Oracle. So family offices have been our largest supporters. We're currently raising a $5 million, well, we're oversubscribed, so we increased it to $7 million convertible note into the Series B. We also are in the process of making a small acquisition of a company that's directly in our space, one that we work with, in fact, one that's a vendor, which is the leader in environmental controls for cell culture, which you'll see in a minute is very important. And we plan to do a $10 to $15 million Series B towards the end of this year. Next slide. So even though cell culture is key to biomedical research, there's been very little innovation in the last 65 years. And when I meet people who say, oh, I would have stayed a researcher if it weren't for cell culture, and they say, I assume it's changed. Well, the answer is it really hasn't. So we still keep cells in plates in an incubator. They're stacked together. To get your cells, you have to move other cells and bump them. Labeling system is often a sticky or a Sharpie, but the problem is it's on the top of the plate and those get mixed up. And we then take these plates to a microscope, and researchers spend 2 to 3 hours a day looking at millions and millions of cells. Do they remember what they see? No. Do they take notes? Some, generally in lab notebooks. And then they put the cells back after moving them across all these environments, stressing them, sometimes mislabeling them, put them back into the incubator. And guess what? There's almost no data that's been collected. So Uber knows far more than we know about cells. Uber knows more about us as passengers than we know about cells. And because of the stress that these cells undergo, moving across all these environments, there's been genetic drift in the cells. So the cells are less and less like the patient model. We're studying these cells because they are supposed to represent us, the patients. But then the amazing thing is we spend billions of dollars studying these cells, billions of dollars on instruments, genomics, and in a quarter of the cases, they're not even labeled correctly. So as a result, different labs get different results. The same lab gets different results over time. And a huge amount of money is spent trying to reconfirm research that's been done. And as the next slide shows, that is not been the case. So there's something called a reproducibility crisis, which is exactly what I just described. And it's recently been studied. It was really because of an article I read about a study that Amgen did that I started Thrive. So Amgen studied 53 landmark oncology studies that were important to them, and they were only able to replicate 10%. There's now been study after study that has at best shown that 50% of experiments are repeatable. And 50% of that problem has been shown to relate to cell culture. If you don't have a reproducible result, you really don't have science. So this crisis, not only leads to a failure rate that's very high in drug discovery, it also puts patients at risk. So in a 4-year period, 125,000 patients in one study were enrolled in trials where the science was later shown to not be repeatable. Next. So as this fellow shows at the bottom of the slide, when an experiment works or doesn't work, we really don't know why, because we don't really have the data. Thank you. So our products are basically trying to solve this problem and really to provide and make up for the lack of imaging. And I want to quote one of our customers who was commenting on the status of products other than Thrive, which is, current tools are failing us because they hurt the cells, only give us 2D single point in time images, and they don't capture contextual data or close to enough data. So what do we need in order to solve this problem? We need lots of data and the right data, basically. It needs to be comprehensive, not biased. So right now, data collection is, a researcher sees something they believe is interesting, and so they take a few digital images. Well, what they think is interesting is biased. There may be other things going on they didn't notice or weren't studying, and they don't have the ability to go back and look. With our system, we take gigabytes of images and collect data in minutes on the cells, and we do it the same way every time, so it is comparable. Comparability means that you can show it to another researcher, and it's useful for them, and they can duplicate it. And our database is dynamic, so we are able to follow cells over time. We're the only instrument that can actually track a single cell over months, because biology is dynamic and the instruments need to be close to the biology. Next slide, please. So we make instruments and software for automated imaging, analytics, documentation, which is incredibly important, so that you can go back and understand what somebody did. And our goal is to make cell culture, tissue culture reproducible. So the same researcher who's chairman of biochemistry at a major medical school who talked about the current failure of tools, after using Thrive, said, Thrive will change the world. Thrive allows us to understand and see living dynamic cells and tissue in 3D over time with remarkable images. He's one of the leading COVID-19 researchers. Pfizer has credited him as one of the principal reasons that they had their vaccine to market first. Another researcher said, for the first time, we have high quality, consistent data to vastly improve experiments and enable machine learning, data mining, and breakthrough insights. Next slide, please. So we have a family of products, which is a little bit unusual for a company at our stage, because we're trying to offer a full set of solutions for small, medium, and large labs. So we have a benchtop system, which is the Cell Assist, which images a single plate at a time in 10 to 15 minutes, produces gigabytes of data. And then we've just introduced Cell Assist 50. We'll actually be formally launching it here in San Francisco next week at the International Society for Stem Cell Research annual conference. And we're able, with the Cell Assist 50, combine our imager with an automated incubator and robotics, and around the clock, every 3 hours, every 4 hours, it images plates of cells, takes millions of images so that somebody can track over time the biology. Most of our customers are generating several terabytes a week. This is far greater than the human genome data that is produced. Nothing's been like this before. Most of our focus now is on software applications and modules that will sell on a subscription basis. And those are basically tools that make it easier for researchers to do a variety of things, such as tracking the single cell or counting cells or studying organoids. Sorry? So, the comment was, or question was, are we creating the next CRISPR? We're closely allied with that. So we help CRISPR, and I'll tell you why. And CRISPR is one of the most important developments in life sciences. The fact that we can edit the genome of a cell means we can create new models of a cell. We can knock out a gene and then study what the effect of it is in cells. And the uptake of CRISPR has been incredible. Probably 40% of all labs do it. But here's the problem with CRISPR that one of our customers told us. So we got a call from Harvard Stem Cell Institute that asked whether or not we could track a single cell over time, and even harder, find a single cell in a well of a plate, which nobody's been able to do. And the reason is that after CRISPR, after engineering, it can be 1.5 to 2 weeks until they can even find those cells. Because without Thrive, you haven't been able to find a single cell in a well. But because we take images at hundreds of focal planes and thousands of images, and we use image processing and AI, we're able to find that cell so that you can actually study it right after genetic engineering and not wait 2 weeks to even know whether the cell has lived. So we enable, it's an example, one of many, where we actually enable some of the biggest workflows that exist. Next slide,
We
please.
got about 10 minutes, Tom.
Okay. So I started to actually talk about this slide, which is one of the key features of what we do, is we are taking thousands of images in minutes of these cell culture plates, which cells grow in. And each of those images is 5 megapixel. And we use very special lighting. Nobody has ever focused on this, using a pun, before Thrive. And we take these images at hundreds of focal planes so that we're really the only instrument that can image organoids or tissues, which are 3D cell structures. This is the fastest growing part of cell culture because it gives us more information. An organoid is basically a collection of cells, and we can study, say, the interaction of a drug, not just on cells sitting at the bottom of a plate, but that are simulating an actual organ. And we're the only instrument, because of all these focal planes, which are adjustable by the user, that we can capture images of the entire organoid. We take the images extremely quickly. Probably, if we decrease it with each release of software, we're probably at 100 milliseconds now. And the importance of that is we're also the only instrument that can image cells in suspension. The importance of that is in immunology, most cells that are studied are not sitting at the bottom, but are floating. And again, we're the only instrument that, because of all these focal planes and the speed, you can actually track those images. The next slide, please. So the main competition for us is basically digital microscopes, which are not nearly as comprehensive. They don't do it the same way every time. You can't track a single cell over time. High percentage are out of focus. We have very advanced algorithms. One of the things that we do is actually plates are not very consistent. So we actually create a physical model of the plate so we can adjust for it and get very, very sharp images. And we document. That's incredibly important. We have a barcode system, which sounds basic, but in cell culture labs, it's revolutionary. And it's the same thing as Cytic, which the company where now 90% of all pap smears use that equipment, which, by the way, has led to pap smears not being required every year, but being required every 3 to 5 years. In those cases as well, in Cytic case as well, there weren't discoveries that need to be made, and there are no discoveries that need to be made at Thrive. We're doing very clever, patentable engineering. We're bringing solutions to an ignored group, and it's not dependent on any kind of a discovery or an FDA-approved discovery. Next slide, please. So I'm just quickly going to go through some examples. So here's how data can make a difference. Remember, I mentioned that on average, a quarter of all cells are mislabeled. Well, this is data from one of the leading institutes. I mentioned their name before. I won't mention the exact name. And for the first time, they're able to see in the data that there's something different about some of the cells, and they're able to look at the outliers. And even though their name is just tops in the industry, like everyone else, they have mislabeling. Some of it may have occurred prior when they bought the cell line, but they can look at the outliers. Next slide, please. So this next slide is, these are images from the person who said that Thrive will change the world. And he's a leading COVID-19 researcher and was able to see for the first time things that were not able to be seen before. So one of the things we do is we study live cells. We study them over a period of time. Currently, the assay that is used in infectious disease research was used, one of the first steps when COVID-19 was discovered, is a viral plaque assay. And what that is you take a culture plate of cells and you put the virus on top of them, and then you wait, well, without Thrive, you wait for 7 or 8 days, and then you wash away the virus, so you have to touch it again, and you stain it. Nobody's been able to image the process prior to the staining. And then of course, once you stain it, you've killed the cells. But we're actually able to see the plaques and the effect without staining. And we're able to see therefore before, and you don't have to kill the cells and after. And that's why he said that they're able to see things that they've never been able to see before. Next slide, please. So this next slide is something called a scratch assay.
About 5 minutes.
I'll just point out that the graph on the left, the importance of that is that each one of those is a drug that was being tested to see the effect on the cells. And the effect that it was looking for was wound healing. So seeing the cells migrate together to improve the wound. And that's all been done basically by humans estimating the speed and how much of the area. For the first time, they were able to screen 100 drugs on live cells and with metrics actually record the effect, which is what each of those lines are. That has never been able to be done before. And which is why I'd say this is a really good example of our bringing tools that will help improve drug discovery. The next slide, this is a single cell. This is from Harvard Stem Cell Institute, tracking a single cell over time. And next image, please. And the next one is basically an organoid. And we have a top of the stack and the bottom of the stack, and you see different things. And there are different dynamics that are going on that we can record. Next slide, please. So I mentioned my co-founder, who's responsible for most of the sequencing instruments that exist. My own background, the most comparable company is Cytic, where we automated cytology labs, which was an imaging problem. I then started a company called Exact Sciences, which has something called Cologuard, where we also basically use data mining to come up with an alternative to colonoscopies. And we have some wonderful board members, including Brock Reeve, who's head of the Harvard Stem Cell Institute. Next slide, please. So I'm not looking to jump out of this company. I look at building value, but investors always ask when we expect an exit, and they should, and they have a right to, and what kind of returns. So the multiples in life science research tools are extraordinary. It's an underfollowed area, but the kinds of multiples that exist are 30x trailing revenues for public companies and 10 to 15 times trailing revenues in M&A. And this is on average, not just picking the best or the worst companies. There are 15 or so likely acquirers. Some of them are mentioned: Fujifilm, Danaher, Thermo, Tecan, Leica, Agilent. Most of them have contacted us, some several times. It's too early for us to sell at this point. Last slide, and this is a summary slide, which is basically, we're looking to transform the field of cell-based experiments where there's very little competition. We're basically doing for live cell biology what sequencing did for genomics, which is to produce lots and lots of data that's accessible and useful for researchers. And this data is critical for drug discovery. We think we have the team to do it. And again, I like quoting customers. With Thrive, we see things we have never been able to see before. And this is what cell culture and cell culture imaging should look like. So if you have any questions, I'd always like the questions I get at Marty's.
Any
events.
questions from the audience here? I have a question. So, you know, it was fascinating when Elon Musk was speaking, I don't know, about a month ago, and they said, well, you know, the reason for Tesla's evaluation is Tesla's really like 5 companies in one, right? And I like to think of Thrive as being more than just an equipment company, right? I mean,
Right.
you've had this interesting evolution of what your platform is. So maybe you can talk about like different areas where you add value.
Yeah, so I should have talked about that in the main slides, which is essentially we're a data company. We're not really a life sciences company. And in fact, the Estee Lauder family, when I told them about my company, said, well, we don't invest in pure life sciences companies. They invest in tech. But within half an hour of the conversation, they said they'd invest. And they realized this is really collecting data. And part of what we're doing is building what will be the largest database in the world, the first one on cell biology, because we maintain licensing rights to all of the images where we're given permission, which so far it's 100% for academic institutions. And we've gotten even some big pharma to do it, but that's okay if big pharma doesn't give us their images because they'll buy the access to the database. The comparison with Tesla or Google Maps is that we're really digitizing an area and collecting lots of data that's useful where data hasn't been collected before. And you look at so many companies have done that in their segment, and nobody has done it in live cell biology, even though it's the basis of all research. So the amount of data that we're producing, because my co-founder developed most of the sequencing instruments, he's used to dealing with terabytes, and we make that all invisible to the researcher. Part of the reason this has never been done, biologists aren't great hardware engineers, software engineers, so we make it very easy for them. And the fact that they aren't, and it makes it very hard actually to build instruments like this, because you bring together so many fields: mechanical engineering, software engineering, optical engineering, databases, image processing.
And the essence on healthcare data companies has been pretty substantial. I mean, I know that, and I think there was an oncological database that sold for billions of dollars a couple of years ago. So I think there's several of those, right? Right.
So interestingly, all of those databases have been on the clinical side. So clinical side is when you study a drug in people. Preclinical is prior to that. And everything's based on the preclinical research. If that's not good, the rest isn't good. And there have not been databases collected because they haven't had the method of collection.
Right. So those have had like 25,000, 50,000 samples. What kind of volume do you anticipate collecting over time?
1,000 times that. We already have more than that from current 15 customers. So, particularly the automated instrument, it's imaging 50 plates at a time, every few minutes, terabytes a day, 50 different cell lines, often within a 96 well plate, there are different cell lines. So we have just a really huge data set. But you really do need big data sets for data mining and for AI. So it's going to be another year or so till it's big enough that we're going to monetize that database.
So it looks like there's 3 business. One business is the equipment business. The second business is the software. First you have the software
Tools.
applied towards the business, towards the equipment, and then a generalized software approach for other tool sets.
That's right.
Right. And then you have the data business.
That's right. That's correct.
Great. Fantastic. Any other questions? We're good. One question.
So the question is, are we cloud-based? And we're not. So we made a mistake of not asking questions quite enough. So, we ended up with, was it new Coke? And they went back to old Coke. The problem is that everyone talks about, oh yeah, we want cloud-based, but big pharma does not, or most biotech companies do not let their data outside of their institution. So even though when we asked researchers what they wanted, they said they wanted it someday. We missed the word someday. So Google actually, a friend of mine runs Google's cloud data and said, by far, the lowest penetration they have is in pharma and biotech. But it will happen. And so, luckily, somebody referenced, even in a ring, you get terabytes of data. So we sell lots of add-on memory so that they don't have to be cloud-based, but also they can map to a server within the organization.
So if you look at like a comparison of like the digital camera in 2020 came out 1.9 fact sets that you collect in the film from new layers of geography of the
Right. So the question is, are we going to have a similar kind of evolution, say, as the iPhone, which started off at 1.2 or 3 megapixels per image, and now is at 10 to 20? So the answer is yes. So I mentioned we now take 100 focal planes, which is like 99 focal planes more than almost every instrument. 6 months ago, we took 20. And so because of the availability, except for recent supply chain, of cheaper and cheaper memory, we're able to expand what we want to collect. So you're absolutely right. There are also many different kinds of imaging modes. We want to incorporate all of those so that we have more and more information. So every instrument, even every software release, adds to it. We're just scanning the begin. It's really, nobody's done it. So there's a lot of value in what we're doing already, but it will get deeper and deeper and the value will increase